Hippocrates has been quoted as saying, "Death sits in the bowels, and bad digestion is the root of all evil," in 400 BC, showing that the importance of the intestines in human health has been long recognized. In the past several decades, most research on the impact of bacteria in the intestinal environment has focused on gastrointestinal pathogens and in the way they cause disease. However, there has recently been a considerable increase in the study of the effects that commensal microbes exert on the mammalian gut. In this presentation, I'd like to talk about the latest research on the subject of the relation between gut microbiota and human health. The gut microbiota exists in complex, multi-species communities, forming a superorganism with the human host. They are together called the holobiont and evolved together for more than 1 billion years. This led to interdependencies forming between them. A large number of diverse microbial species reside in the distal gastrointestinal tract. There is approximately 1 bacterial cell in our intestines for each of our human cells. There are almost 30 trillion bacteria. Bacteria have been found in the placentas of healthy mothers, in amniotic fluid, and in meconium. Delivery method influences the newborn's gut microbiota, making it similar to their mother. As the host human ages, diversity of their microbiota increases, reaching maximum suitability in adulthood. During this maturation, every person's microbiota becomes unique to them. In the elderly, gut microbiota becomes unstable and less diverse. This is associated with coexistent conditions and age-related declines in immunocompetence. In this graph, we can see that bacterial diversity and functions decrease with age and diet, over the age and diet. It gradually increases over the first years of life and slowly decreases over the end of life. As microbiota is such an integral part of the human holobiont, it is influenced by many factors, especially gender, age, diet, antimicrobial agents, and stool consistency have been shown to exert large effects on the gut microbiota. Interestingly, the host genome is associated with heritable bacteria. Christensen's Lycia is an example of this phenomenon, as shown by a study on stool samples from twins in the UK. Monozygotic twins had similar concentrations of Christensen's Lycia. When administered to mice, it reduced weight gain. Also, single nucleotide polymorphisms are associated with proliferation of specific bacteria in people with inflammatory bowel disease. In this graph, we can see that microbiome is tightly interconnected with IBD pathogenesis. 4 of the microbes here are associated with IBD pathogenesis. Immunity is also a big factor influencing microbiota. Sensing of microbes by regulatory T cells promotes mucosal tolerance and enforces commensalism. In another study, 3 distinct microbiota states were described in babies 1 month of age, and babies with one of these states had a higher relative risk of allergy in 2 years of age and asthma in 4 years of age. This high-risk state has been associated with high interleukin-4 concentrations. This may be indicating that early life microbiota can precede childhood disease development. A diet high in meat can increase bile metabolizing microbes, which is associated with inflammatory bowel disease like Crohn's disease and ulcerative colitis. It has also been reported that people have different responses to identical meals. Personalized meals can help control blood glucose levels. Oral or nasal exposure of mice has been shown to prevent airway sensitization and asthma. In a clinical trial, researchers used a machine learning software and data from various surveys and immunological and biochemical tests from subjects, approximately 100 subjects, and associated them with specific diets and measured their postprandial glucose levels. When they used these data to modify their diets, to personalize their diets for each individual, they achieved better glucose control, postprandial glucose control in each of the subjects. Vibrio cholerae dominates fecal bacterial communities during infection. Recovery is associated with restoration of pre-infection gut microbiota resembling of infancy. Gut microbiota undertakes many critical roles in human physiology. Disruption in each of these roles is ground for disease. Please notice the wide variety of functions. Gut microbiota dysbiosis, imbalances in the composition and function of these intestinal microbes, is associated with diseases ranging from localized gastroenterologic disorders to neurologic, psychiatric, respiratory, metabolic, hepatic, and cardiovascular illnesses in human and rodent studies. Research on these subjects is difficult because of the intra-individual variability of the microbiota caused by a myriad of influencing factors. Human obesity, kwashiorkor, childhood asthma, massive weight loss after bariatric surgery, and the insulin-resistant state of third trimester pregnancy has been shown to have causal relationships with specific dysbiotic states of gut microbiota. Rodent studies have shown that specific bacteria may cause insulin resistance. Transplantation of fecal microbiota from healthy lean humans, as will be discussed by the next speaker, to obese patients with insulin resistance improved whole body insulin sensitivity. A common ground hypothesis has been proposed. It suggests that the factors influencing the microbiota increase gut permeability or mucosal inflammation. By doing so, they favor opportunistic microbe intervention in genetically susceptible individuals. This change in microbiota results in polygenic disorder, therefore the disease phenotype. In this graph, I described that many chronic disorders are thought to result from a relation between dysbiosis and host genetics. Together, they come together and form many of the chronic illnesses we know, and many bacterial infections are in fact result from host genetic susceptibility to them. These are the main areas we are currently looking at for using microbiota in disease treatment or prevention. The boxes in red show rodent studies, and the boxes in blue show human clinical studies. In treatment of antibiotic-resistant Clostridium difficile, fecal microbial transplantation results 90% of recurrent diarrhea cases. This finding proves that microbiota from healthy donors can correct the dysbiosis, and fecal microbial transplantation is a viable medical therapy. Probiotic medicines can be prepared specifically for the patients and their gut microbiota. Studies have shown that commensals produce trimethylamine from phosphatidylcholine and L-carnitine. Trimethylamine then forms trimethylamine N oxide, TMAO. TMAO is associated with atherosclerosis in humans and animals. 3,3-dimethyl monobutanol administration inhibits commensal trimethylamine production and protects against atherosclerosis despite the pro-atherosclerosis diet. Here's a diagram of the study. The gut microbes intake phosphatidylcholine, metabolize it, and make it into turn it into trimethylamine, which is proven to cause atherosclerosis in patients. By administrating 3,3-dimethyl-1-butanol, we inhibit their uptake of choline and block their production of trimethylamine, and therefore block atherosclerosis pathogenesis. In this slide and the next, we can see that by inhibiting microbial uptake of choline, we can prevent atherosclerotic lesions from forming in mice. This is a microscopical slide. You can see that relative to control groups, the groups with DMB applied, 3,3-dimethyl-1-butanol, there are far fewer lesions of atherosclerosis in the experiment group. This is another slide. This is from tricuspid valves of the heart of mice. You can see that choline is associated with radical atherosclerotic plaque formation, and in the experiment group with DMB applied, there are almost no atherosclerotic plaques. There are almost no atherosclerotic plaques in experiment group. Gut microbiota is also being explored in oncology. Orally administered bifidobacterium modulates dendritic cell activation, which in turn improves tumor-specific CD8-positive T cell function. Bifidobacterium supplementation improved tumor control to the same degree as anti-programmed cell death ligand 1 therapy in an animal model, and combination of both almost completely eliminated tumor expansion. In this table, we can see that cancer and microbiota have a relationship on both local and systemic levels. There is a tight relationship between human immune system disorders and gut microbiota. A mixture of 17 human Clostridium strains has been shown to diminish the severity of experimentally induced allergic colitis in rodents through mechanisms that promote the expansion and activity of Treg cells, although it remains to be determined which bacterial molecules mediate these effects. In the field of metabolism, it has been found that E. coli secretes CLPD, which is a chaperone protein and alpha-melanocyte stimulating hormone analog, affect food intake and meal patterns in rodents. E. coli proteins stimulate intestinal glucagon-like peptide production, which is a potent anti-hyperglycemic hormone, peptide YY production, which is produced in the ileum in response to feeding, and activate anorexigenic pathways in the brain, inducing satiety. In another study, neonatal probiotic supplementation was associated with a 60% reduction in the risk of pancreatic islet autoimmunity before school age. E. coli was genetically manipulated to biosynthesize precursors of the anorexigenic and acylethanolamides. When administered to obese mice on a high-fat diet, resulted in lowered food intake, increased basal metabolic rate, and loss of adiposity. The effects did not disappear for 4 weeks after cessation of supplementation. Bacteroides fragilis administration ameliorated experimentally induced anxiety in mice. As a member of normal colon flora, it also diminished behavioral abnormalities and restored mucosal barrier integrity. In most studies, germ-free mice show a decreased anxiety phenotype, but exacerbated response to stress and have numerous neurochemical disturbances. Chronic stress significantly alters intestinal microbiota composition, primarily reducing lactobacillus count. Reactive oxygen species produced by lactobacilli can inhibit kynurenin's metabolism, which can negatively impact the brain when dysregulated. It has also been shown that Lactobacillus reuteri can modulate GABA receptor expression in the central nervous system via the vagus nerve. Therapeutic administration of Lactobacillus reuteri to stressed mice improves metabolic homeostasis and corrects stress-induced despair behaviors. Kynurenine metabolites, kynurenic acid and quinolinic acid, form a balance of neuroprotection and excitotoxicity over glutamate and MDA receptors. Peripherally, kynurenine is metabolized into quinolinic acid, which is responsible for excitotoxicity in the brain, and in glial tissue, it is metabolized into quinurenic acid, which antagonizes quinolinic acids in excitotoxicity and protecting the brain. Quinolinic acid is associated with Alzheimer's disease and proven to cause the disease, and the blockage of quinolinic acid by quinurenic acid decreases the rate of disease progression. In conclusion, microbiota is becoming one of the main targets in research for therapeutics. While murine studies show promise, most of them weren't tested for treatment in humans, and they were performed under highly controlled laboratory conditions. How well these findings will translate to medical practice remains to be seen. Because every human's microbiota is still different, personalized symbiotics may hold potential for therapy. Thank you.